Three main blood vessels: arteries, veins, and capillaries, starting with arteries. So these transport oxygenated blood that carries oxygen, away from the heart under high pressures. Very important detail. If you ask for a definition, probably not, maybe on the multiple choice. All of this except the pulmonary artery, which carries deoxygenated blood towards the heart. Now, arterioles is the word for tiny arteries that connect to capillaries, while the aorta in the heart is the largest artery in the entire body. Okay, let's have a look at the structure of arteries on a deeper level. Keyword here: in the artery wall, there's something called elastin, which is going to stretch it to accommodate for the increased volume of blood during contraction. So when the ventricles of the heart contract, there's going to be more blood moving through the arteries, and so these are going to need to respond through stretching. Okay, with their elastic walls, they contain elastin. Now these are going to recoil as the heart relaxes. Okay, and this is going to push your blood forward. Very important. Now this elasticity reduces fluctuations in blood pressure, so it's going to maintain a relatively stable blood pressure. Okay, so the artery doesn't burst. Now smooth muscle, I'm going to underline it here. Smooth muscle, so the muscle found in your blood vessels. Okay, skeletal muscle would be the muscles found attached to your bones. Okay, so the biceps, the triceps, the muscles on your arm and your legs. These are skeletal muscles, and now we're looking at smooth muscles. Okay, so these are going to regulate the diameter of the lumen. The lumen is basically just the hole inside the artery, this thing here. Okay, and the diameter, you know, from maths, this would be the diameter. Okay, so it's going to regulate the diameter. It's going to make it bigger or smaller depending on the part of the body and whether it's in vasoconstriction or vasodilation. Vasoconstriction is when the blood vessel gets tighter, so it decreases the diameter of the lumen, and vasodilation is when it dilates. The diameter of the lumen gets bigger. Okay. Now, let's move on to the three layers of the artery. First layer, the tunica intima. Okay. It contains the endothelium layer, which creates a smooth, friction-reducing lining. Ooh. Next one, tunica media, is made of the smooth muscle we just talked about and the elastin, the elastic fibers. And now the outer layer, the tunica adventitia. It is made of mostly collagen fibers, which protect the artery and anchor it, so, you know, like an anchor, stick it to surrounding structures. We know that arteries carry oxygenated blood away from the heart, and veins do the opposite. They carry deoxygenated blood towards the heart, except, of course, the pulmonary vein, which carries oxygenated blood to the heart. Okay, it is still to the heart, but this time is going to be oxygenated when it comes to the pulmonary vein. Okay, let's look at the structure of veins. These veins have a flexible wall to allow contraction of skeletal muscle this time, so muscle attached to bones, moving the blood. They also have valves, okay, and these prevent the backflow of blood. You might have seen in heart diagrams, blood vessel diagrams, these little wings or whatever they're called. These are the valves, okay. So they prevent blood moving backwards, so it only moves in the direction that your body wants, okay? They have the same three layers as arteries, but this time they have a thinner wall relative to the diameter of the lumen. We're going to look at a diagram that perfectly represents this, okay, which is high. The diameter of the lumen is high in arteries, but not in veins, okay? And they also have less smooth muscle and less elastic tissue in the tunica media, so in the middle layer. Media for medium, okay? Middle. Now let's compare the diameter of the lumen on both, okay, and the size of both. Now we can see that the vena cava, which is a vein around your heart, is actually larger, okay? It's larger, and so the diameter of the lumen is also larger. But the layers are actually thinner than the aorta, which is an artery, okay, which has a smaller lumen, but the layers are still thick. Look at how thick the layers are, okay? And this is because it wants to sustain more blood pressure, because it carries blood that has just been pumped by the heart, because it carries the oxygenated blood away from the heart. All right, capillaries. Let's have a look. So they facilitate the exchange of substances between the blood and the internal, so the fluid in your body, and the external environment, being like the air. So for example, oxygen goes from your capillaries into your cells, okay, for aerobic respiration. And in exchange, as the function of capillaries suggests, in exchange, CO2 goes out from these cells out to the capillaries, and from the capillaries it goes out, okay, to the lungs, and then it's expelled out into the external environment. So now we're going to look at an example of an exception to this. The function, the structure of capillaries and comparing veins and arteries in a microscope diagram. So going back to the exception. So this is an exception because it's a one-way exchange. Okay, so you give a substance, but you don't get a substance back. So this is in the small intestine, where capillaries absorb nutrients, but they're not going to expel them back out. That doesn't make sense, okay? So that's the exception you need to know. Okay, structure of a capillary. First thing, their walls are one cell thick, only one cell thick. Why? To decrease diffusion distance. Remember that their function is the exchange of substances, and this is done by diffusion, so we want as much diffusion as possible. So decreasing the diffusion distance increases the rate of diffusion. Very important. Fenestrations. So there's going to be gaps, they're labeled here, called fenestrations, and capillaries with these fenestrations are usually found in organs, with high metabolism. So metabolism, as in a lot of chemical reactions are going on. I'll give you an example. The liver would be one. Okay. The extracellular matrix, also known as the basement membrane. Okay. This thing surrounding the capillary provides structural supports and controls the exchange of which materials are entering or not entering. That's why we call it the basement membrane as well, like cell membrane, but it's not exactly a cell membrane. Now, most capillaries are continuous, allowing for selective exchange of small molecules and ions. Selective as in selecting which molecules and ions enter or leave. Okay. Now, their lumens have a diameter of 5 to 10 micrometers. So that's pretty large compared to arteries. Well, their wall thickness to diameter ratio is different to the arteries, certainly. So they're highly branched, okay, as you can see. And this means that they have a very surface area to volume ratio, okay? There's a lot of surface area and little volume. That's why I said the walls are very thin and they have a lot of surface area, okay? And this, again, is very important to slow the flow of blood and give more time for the exchange of substances. When looking at a microscope comparison of veins and arteries, we can see how this vein right here, how veins in general, have a very large lumen diameter. Remember, the lumen is the hole, compared to arteries, which have a smaller lumen diameter relative to their thickness. Okay, we know that arteries have very thick walls, as you can see, compared to veins, and especially capillaries, which are only 1 cell thick. Veins aren't 1 cell thick, but they still have short walls. Okay, so the arteries have these larger walls. Why? To sustain the pressure that comes from receiving the oxygenated blood right from the heart. Okay, so it's very important to recognize the cell thickness, sorry, the wall thickness and the diameter of the lumen when trying to identify or being asked to identify whether it's a vein or an artery. Okay, now this is another little thing inside this topic. You need to know how to measure your pulse rate. Okay, you're using your your heart rate. Remember that the most professional way of doing this would be to use a stethoscope. So when the doctor puts this device on your chest, okay, around the sternum, this would be a professional way. Also remember that you probably know this already, but it's worth mentioning. When you put your fingers around your radial artery, okay, around the hand, that's another way of measuring your pulse rate, though not as accurate as a stethoscope. Moving on to a, whoa, I should have added animations. I apologize, but I'm gonna use this red cat thingy to guide you. So we're talking about coronary heart disease. So first let's understand the importance of the coronary artery right here. Okay, so it's by the heart, as you can see, and it's going to provide uh, essential nutrients and oxygen to your heart muscles. Okay, and it branches off the aorta, which is, remember, the largest artery in the body. Okay, so in the case of having a cholesterol plaque in this artery, okay, this yellow thing right here, that's going to make this smaller, this coronary artery have a smaller area around here, this is going to be a problem, okay? Why? Well, it can lead to a type of cardiovascular disease called coronary heart disease, or CHD. Okay, and this is caused by the occlusion, keyword, just remember that it means blockage, of these coronary arteries by these cholesterol plaques, okay, which eventually harden into something called an atherosclerotic plaque, okay? We're gonna look at these in a second. So this coronary heart disease leads to ischemia or ischemia. I'm not exactly sure how it's pronounced, which is basically the word for lack of blood flow. Why is there a lack of blood flow? Well, because the artery is blocked, or not fully blocked, but partially blocked, okay? And this is due to the atherosclerotic plaques. So remember, cholesterol plaque turns into atherosclerotic plaque. These rupture and form blood clots or thrombus. Go back to the previous diagram. Thrombus in this coronary artery. And this leads to another condition called angina, which is the word for shortness of breath, fatigue, and pain because the heart is not getting enough nutrients and oxygen. And this can eventually lead to myocardial infarction, which is a heart attack. Any questions can go in the comments. I think that was a pretty decent explanation. Now we've done transport in animals. Let's look at transport in plants. First thing, you've probably heard of the xylem, GCC. It transports water and dissolved minerals, and it does so unidirectionally. Okay, so in only one direction. Now, the transport of water and dissolved minerals by the xylem is possible thanks to transpiration, okay, which is the loss of water from aerial parts of the plant through tiny pores called stomata, which are on the leaves, okay? So as water evaporates via the stomata, this creates a negative pressure potential, okay? Why does this cause a negative pressure potential? Well, because there's less water potential outside the stomata compared to inside the leaf. And this makes sense, right? Because inside the leaf, there's all the water, and in the air outside the leaf, there's not as much water. So there's lower water potential outside, okay? Now, water is pulled out of the xylem, okay? And this causes water to pull up via the xylem as capillary action. So how do I explain this? It's pulled out of the xylem, and this causes water underneath the xylem to be pulled up through the xylem, okay? And this is done through capillary action. Hope that makes sense. And this happens due to the cohesive and adhesive properties of water. Remember, this is A1.1, okay, I have a video on that as well, but let's recap. Cohesive means that the molecules can bond with the same molecule, so water can form bonds with water, we know that. And adhesive is that it can form bonds with different molecules, so water can form bonds with other molecules as well, okay, not just itself. And this is caused by the polarity, in this case dipolarity of water. Again, this is A1.1, okay? So going back, water is pulled out of the xylem, and this causes water underneath to be pulled up through the xylem through capillary action, and this draws more water into the xylem from the soil, okay? So essentially, water being pulled out of the xylem causes like a chain reaction, right? Where the water underneath keeps getting pulled up, okay? And this is the negative pressure potential that we've just talked about. Now we talked about the xylem, let's mention the phloem, okay, which facilitates the transport of carbon compounds, okay, including sugars such as fructose, glucose, and amino acids, okay, which make up proteins. So I always confuse these, but sugars and amino acids are not dissolved minerals, okay? The keyword dissolved is important here because the minerals in the xylem are transported inside the water because they're dissolved inside the water, okay? Now, the sugars and amino acids, they are not dissolved. They are just carried, okay? Very cool diagram. Let's attempt to explain it, okay? So, this is the outside air. The outside air has less water potential than the leaves, okay? This causes the negative pressure gradient, okay? And this causes the chain reaction of how water gets pulled out of the xylem. This causes the water underneath the xylem to be pulled up through the xylem, and this causes the water in the soil to be pulled up inside to the roots, okay? Now, in this step where water is pulled up via the xylem, okay, let's have a deeper look into cohesion and adhesion. So remember, cohesion is the bonds within, between water molecules, okay? This is hydrogen bonding. Adhesion is also hydrogen bonding, right? So cohesion is between water molecules, and adhesion is the water molecules binding to the cell walls of the xylem, okay? And apart from that, this was just revision of what I said before, just with the diagram. Let's move on. Okay, looking specifically into the xylem, different characteristics of it Okay, it's made of dead specialized cells. These are the cells that water adheres to. Okay, the adhesion happens between water and these cells, and that's how it moves up. Okay, now the wall of the xylem vessel is strengthened and impermeable, sorry, so water can't go through it. Okay, of course we don't want water to go through it because we want the water to go up. Okay, it's also strong to um, manage the osmotic pressure. Okay, and this is all thanks to something called lignin. Okay, so the lignin allows the impermeability and the strength. Now, it's also hollow and thin. Why? Well, we know that the hollower and thinner a tube is, as well as the longer it is, the more capillary action there's going to be. And this is part of a 1.1 as well. The old syllabus, I think, but it's still relevant, I'd say. Good to know. Now, the lumen of the xylem contains no cytoplasm. Okay. The pits, these yellow circles you can see here, are for conducting the water. The lignin deposition is in the form of spiral thickening. So this red spiral you can see here is the lignin deposition. And I say we move on to the structure of the stem. A dicotyledonous plant is a flowering plant with two or embryonic leaves, also known as cotyledons, in its seeds. Remember, di always stands for two. Okay. Now let's look at the structure of the stem in these plants, because it contains xylem, phloem, and a bunch of other structures that we need to identify on microscopes. First thing, the outer layer of the stem is called the epidermis, which contains epidermal cells. These have what's called a waxy cuticle made of lipids that prevents water loss from the stem. Okay, we don't want water to be lost. We want water to go up the stem, except in areas with stomata. Okay, stomata isn't only in the leaves. Okay, we want some transpiration through the stem, but they're going to be mostly in the leaves. That's where we want the transpiration to happen. Okay. Now, these epidermal cells are also found in the roots, okay, where they're going to absorb the water and minerals from the soil. And these epidermal cells also contain root hairs to increase surface area for efficient osmosis and active transport. Osmosis for pulling the water from the soil to the roots, and active transport for pulling the nutrients from the soil to the roots. Okay. Now, remember that the area in the middle of the stem is the pith. Okay. The second outer layer is the cortex, or the layer that's closest to the epidermis is the cortex. Okay. And then there's the cambium, which is the line at which the xylem and phloem intersect. Okay. Now, very important, the xylem is in the middle and the phloem surrounds the xylem. Based on this, I have this mini quiz here. Okay. You have four options. Some of them can repeat themselves twice. Okay. This is same thing here. And you need to fill in these gaps based on these options. And I'm going to give you the answer in 1 second. Okay. So you can start writing. Pause the video right now. Start writing. Let's see if you get it right. So I hope this diagram is visible. You have the diagram of a microscope. Two different diagrams. Okay, label. Pause right now. Label. Showing answers in 3, 2, 1. Let's go. Easy 7 out of 7. Yes, sir. Now, as I said before, the xylem is around the middle. The phloem surrounds the xylem. Okay. You know, it's actually called xylem because it forms like an X shape. Can you see it? Like this is an X shape. It does like an X shape like this. That's why it's called the xylem, I think. Anyways, now the cortex is the next layer that goes before the epidermis. The whole thing containing the xylem and phloem is called the vascular bundle. Remember that. And then we need to identify the phloem, xylem, and cortex in this other diagram. That's it for this topic.